I started off by talking about old stuff, I guess. And ever since energy and protein were discovered as nutritional characteristics of feeds, we've been doing ration formulation according to this model of knowing what the energy and protein supplies from the feed are, and then calculating a requirement from some performance variables on the animal, and get a balance then on the energy and protein. Is it positive or negative? And we want it to be, slightly positive. And as our nutritional knowledge has gotten better, we've gotten more sophisticated at describing the nutrients in feed and what types of carbohydrates there are, what types of proteins, and the amino acid content of the protein, and a lot more sophisticated stuff. But we're still doing the same balance of the feed supply against a requirement from the animal. And ultimately, where we want to get to is to be able to predict the entire response to the nutrient supply. Because if we can do that, then we can optimize various things. We don't have to just be stuck to a certain requirement, but we can optimize like returns over feed cost, or like nutrient capture in feed, in product, or, environmental variables like that, or animal health characteristics. So our lab has been working in this direction, trying to predict the nutrient response, and then specifically around amino acids. The essential amino acids, their supply can be manipulated, and we want to be able to predict how much the milk component yields will change to whatever that supply is. And, the hypothesis that's been used for the past like 80 years or so to do this kind of prediction is the law of the minimum. And I'm glad that Kate and James showed this in their talk last month, the barrel stave representing the law of the minimum. And so you should expect that you'll only get a response to lysine according to this barrel that's depicted here. But actually, in some of the very first experiments that were done on dairy cows to try and identify what the potentially limiting amino acids are, Chuck Schwab did a whole bunch of different combinations of essential amino acids and found that methionine and lysine stimulated milk protein yield, but in those same cows on that same base diet, arginine and histidine also stimulated milk protein yield. And then if we look further at, experiments they did down the line where there's methionine and lysine in all of these treatments, so looking for the next limiting amino acid, isoleucine and valine infusion gave a milk protein yield response, so did threonine. So these results totally defy the law of the minimum. And that was in 1976. You might think, oh, don't you have anything more recent than that? And actually I do. Not too many people really try and test out the hypothesis underlying. We just assume that the barrel stave is the way things work. But I was fortunate to work with Peter Yoder, a PhD student, with Mark Hannigan at Virginia Tech. And he did this very carefully designed and executed experiment with infusing methionine, lysine, and histidine into cows that were actually very high producing. So they were like a little bit over 50 kilos a day of milk production. And so very high milk protein yields that are being shown here. But methionine, lysine, and histidine stimulated milk protein yield. So did isoleucine and leucine, again defying that law of the minimum. And the two groups together were additive, so you got an even further increase in milk protein yield. So my thinking is that there's got to be something else regulating milk protein synthesis than what we've traditionally been thinking about in terms of the law of the minimum. And so that there's the mTOR signaling and EIF2 signaling that happen, and there's phosphorylation of these three primary translation initiation factors that changes the rate of protein synthesis through a signaling cascade that amino acids can be involved in. So they will affect mTOR, they'll affect the EIF2 phosphorylation state. So our thinking is that the law of the minimum is being violated by differences in essential amino acids and how potent a signal they are for mTOR in the translation activation. Like leucine is a potent mTOR signal. And so different amino acids could have different effects. I'm not talking about a kinetic effect of the amino acid concentration. I'm talking about a signaling effect through these various pathways. So we've studied them for a while. This is some work that Sergio Burgos did as a PhD student here. These are incubations of mammary epithelial cells with amino acids or with lactogenic hormones or a combination of them. And those various factors stimulate mTOR. These are the markers of the mTOR signaling. And together, the amino acids and the lactogenic hormones stimulate mTOR even further. And we see the same thing in the protein synthesis, that as mTOR is stimulated, so is the protein synthesis. And actually, Mark Hannigan's group down at Virginia Tech, they did some infusions into cows, short term, so just a day and a half. And they infused either casein or starch, and they got a milk protein yield to the combination of those two nutrient infusions. And that's the one that gave them an mTOR activation signal as well. So this pathway is operating in the mammary glands, is responsive to the nutrient supply. But we've done some further experiments with longer term infusions, still fairly short nutritionally, but after 5 days of infusion, this is an experiment that John Dolman did as a postdoc. So either infusing all the essential amino acids or mixtures that are lacking one single essential amino acid. And with those amino acid infusions, we see that the mammary mTOR signaling is really activated. In a couple places we see it here. But the milk protein yields don't change. So there's a disconnect between like that translation signaling and what the milk protein yields are. So we don't have a complete answer to everything with this yet. And actually the EIF2 here may have been an explanation as why we looked at it, you know, with amino acid imbalances or deficiencies. It's supposed to be an increase in phosphorylation of EIF2 that makes the protein synthesis go down, but we didn't see that. So we're missing something here.
Hey, John. Sorry to interrupt you.
Oh, hey, Boning Lee. I'm glad you
Hey,
stopped by. I hope you can have some information to help me out with my problem here.
yeah, of course. Well, I think you mentioned a lot of results that relate to the translation part. Have you ever thought about the transcriptional control by the amino acid?
No, I don't. I don't think I've thought about it enough. I bet you're going to tell me some stuff.
Yep. So, well, today I want to focus on the transcriptional factor to discuss with you guys about the how the amino acid can affect the milk synthesis. Well, first things first. So, what is a transcription factor? The transcriptional factor is a protein that controls the rates of the transcription by binding to a specific DNA sequence. The function of the transcriptional factor is to regulate the genes in order to make sure that they are expressing the right cell at the right time and in the right amount of time throughout the life in the cell. The transcription functions involving the cell division, cell growth, and cell death. So, why we should pay attention to the transcription factors? We have been studying mechanism which amino acid profile and supply could affect the milk protein production. Through the mTOR and ISR signal pathway, we have been noticed that sometimes other aspects of the milk synthesis, like the milk fat, are affected by the amino acid supply. We also realize that the memory epithelium cell proliferation, differentiation, and death are important determinants of the milk production. When the mTOR and ISR pathway are activated, change in the gene transcription occur. We want to find out which transcription factors are activated by the amino acid and which genes are the affected transcription. So we want to design a research trial that could explore the relationship behind the amino acid and transcription factors. The objective of this research is to find one or several key transcription factors which have the significant effects on the milk synthesis. Well, so how are we going to achieve this research goal? The first method in my head is to use RNA sequence. It is a great method to know all the potential genes, but the disadvantage is also super clear. It will give us like too many genes results and too many informations for the statistical method. Another method occurred to me is the DNA pull down. It is a very good method to know the potential genes, but it also requires one single candidate genes to reflect the outcome, which we don't have that. Well, finally, we decide to hypothesize important transcription factors and evaluate the outcome with the silencing RNA. The great part of this method is we could make changes along the way. Well, this is a summary figure that shows how the amino acid potentially affect the transcription factors. Well, I call this potentially because we summarize this out by finding researchers in all kinds of animals. I'm not going to go into super details about this figure, but as you can see here, the bottom are the 8 important transcription factors that we picked out, which are the SREBF1, MYC, ATF4, FOS, Jun, EGR1, and ATF6. Well, these two transcription factors, SREBF1 and ATF4, has been well researched in human and non-ruminal animals. The SREBF1 is related to the liposynthetic pathway, and the ATF4 is part of the ISR pathway and mTOR pathway. The ATF4 is also related to the amino acid transport and cell apoptosis. So what experiments going to look like? We decided to do the in vitro trial first by using the primary cell culture in memory epithelium cells. The treatments are 4 levels of the single amino acid supplement and keep the rest of the amino acid in the normal level. They are like 25% of the normal amino acid level, normal amino acid level 2 times and 4 times of the amino acid levels. So this is the timeline of the experiment. First, I will collect the organoid from the slaughter cows of the memory, thanks to Carolina's trial. Then I grow the memory epithelium cells from the organoid and passage them into the 6-well Petri dish to get enough cells for the experiment. After that, I grow the cell to reach 100% confluency in the 6 wells and differentiated the cell. When all the cells in the Petri dish get differentiated, I knock down the certain transcription factors in half of the wells. Finally, I do the amino acid treatment and harvest the cell for PCR and other analysis. Yep, so that should be everything for me today.
All right. Thanks a lot, Boreng. That really covers a lot more detail around the memory side of things. And another thing that I'm concerned about in terms of predicting amino acid responses is that the amino acids that go to the mammary glands, that's not the only place where the absorbed amino acid can go. So there's exchange in and out of body pools, and then there's catabolism as well. So this partitioning might have an effect. And to show you what I'm talking about, let me give an example. You know, the branch chain amino acids include leucine, which is, like I said, a potent activator of mTOR, and you've mentioned that. But it can do it in the mammary glands, but also in the muscle as well, and stimulate its own utilization for protein deposition in muscle. And then we also know that the essential amino acids, they stimulate their own catabolism as well, expression of the catabolic enzymes, wherever that's happening. And so, you know, to illustrate, here's an experiment that Rochelle Curtis did as a PhD student here, infusing glucose into cows. It was 1 kilo a day, and these are the amino acid concentrations that were observed. And glucose did cause the arginine and lysine concentrations to go down a bit, but it really affected the branch chain amino acid concentrations a lot. And we see this, it could be in a cow, it could be a human. You know, if you give them glucose, you'll see that the branch chain amino acid concentrations are really affected by that. So we were wondering what happens if we replace those, you know. So we did an experiment, this is still Rochelle's, infusing glucose at 1 kilo a day, but then adding in branch chain amino acids at 75 or 150 grams a day. We weren't really sure how much we needed to get the concentrations back to normal. But here you see the blue bars are the branch chain amino acid concentrations. They drop with the glucose, and then we replace them. So actually 75 grams a day was sufficient to replace, you know, put the branch chains back to normal. But we got all 3 of the levels to do our stats, our linear effect on. And what was surprising was that as the branch chain amino acid concentrations increased, that the actual output of milk protein decreased, even though the uptake of branch chain amino acids was higher and was increased by the infusions that we gave. But also, you know, the utilization of the branch chains elsewhere in the body was increased. So this is the remainder, taking what was absorbed, calculated to be absorbed, subtracting off what was taken up by the mammary glands, and the remainder is also going somewhere else. So this is into muscle or catabolism. And then when we look at the non-branch chain essential amino acid concentration, so, you know, lysine, methionine, histidine, arginine, all that stuff, We saw that as the branched chain amino acid concentrations went up, that the non-branched chain essential concentrations went down. And so their output in milk went down, their uptake by the mammary glands went down with the additional branched chains, but their utilization elsewhere in the body was increased. So it looks like the branched chains stimulated utilization of all of the essential amino acids into non-mammary places. And so then that's another reason why this law of the minimum can be violated. It's just this redistribution between body pools.
Hey, John, I have a question. You talked
Okay.
about chain amino acids influencing partitioning, but what about other essential amino acids such as arginine?
Okay, I'm all ears. I'd love to hear about it.
So my research project is interested in understanding how the semi-essential amino acid arginine affects the gene expression between the mammary and muscle tissue. So why are we looking at arginine? Well, it is a highly versatile amino acid and acts as a precursor for many physiological functions. Arginine can convert to ornithine, and ornithine can be used for non-essential amino acid synthesis and polyamine synthesis, where polyamines are important for cell differentiation and cell survival. Jumping down to the bottom arrow, arginine can also convert to citrulline and nitric oxide. Citrulline is able to recycle back to arginine, while nitric oxide acts as a potent vasodilator and regulates the blood flow and blood pressure. As well, it's involved in cell signaling and has a role in the immune system. Arginine is known to be involved in nitrogen metabolism, as it's a key role in the urea cycle found in the liver, but it also acts as a nitrogen donor for transamination reactions during the synthesis of non-essential amino acids. Research has found that arginine has a role in the activation of mTOR by cellular arginine sensors. And in regard to milk protein synthesis, arginine is a big precursor for endogenous synthesis of non-essential amino acids. Research has found that arginine has a role in the production of creatine, and this plays a role in energy metabolism. And lastly, arginine has been shown to influence hormone secretion, such as growth hormone, insulin-like growth factor 1, and prolactin. As well, some studies have shown that supplemented arginine improves reproduction and growth rates. So the goals of this project are to try to answer questions including how does arginine affect gene expression within certain metabolic pathways between these two different tissues? Of these two tissues, which is more sensitive to arginine? And how does the supply of arginine influence the gene expression? The animal phase was conducted at Agriculture and Agri-Food Canada Dairy and Swine Research and Development Centre in Sherbrooke, Quebec, where we used 6 rumen cannulated multi-paris Holstein cows that were randomly assigned to a 3x3 replicated Latin square design with 14-day periods. The treatments were continuous abomasal amino acid infusions that included 0, 49.2, and 98.4 grams per day of arginine. The cows were milked twice a day, and abomasal infusion lines were checked daily. They were fed 12 times per day from automatic feeders that gave an equal portion of the diet every 2 hours, and the diet consisted of mature hay, silage, and concentrate, which provided 100% net energy and 75% protein requirements. At the end of each period, we collected mammary and longissimus dorsi biopsy samples from the 6 cows. These samples are being used for gene expression analysis. After sedation and a local anesthetic, a trocort was used to collect the mammary tissue, and a biopsy pen was used to collect the longissimus dorsi. And we collected about 700 to 800 milligrams of tissue per biopsy, and it was washed in saline and then snap frozen in liquid nitrogen. Gene expression analysis by RT-qPCR will aim to understand arginine's role in metabolism and how it may affect gene expression related to topics such as the arginine metabolic pathway, which I'll cover in a little bit more detail in the next slides. Blood flow regulation, as arginine acts as a precursor for vasodilation, mainly through nitric oxide production by nitric oxide synthase. cellular amino acid transporters, protein synthesis and regulation, as arginine acts as an mTOR activator through castor proteins, and SLC38A9 transporter for 2 examples, cellular signaling such as the JAK-STAT5 pathway, and genes associated with the cell cycle and to do with cell proliferation, apoptosis, and survival. And this is the arginine metabolic diagram I've been creating, and it highlights some of the many pathways arginine is involved in. To look at it a bit more closely, arginine can produce ornithine by the arginase enzymes, both present in the mitochondria and cytosol. Cytosolic ornithine can lead to polyamine synthesis by enzymes including ornithine decarboxylase, pyrimidine synthase, and pyrimine synthase. Whereas mitochondrial ornithine can lead to the generation of non-essential amino acids by enzymes such as ornithine aminotransferase, pyrroline 5-carboxylate reductase, and pyrroline 5-carboxylate dehydrogenase, just to name a few. And the last pathway I will share is arginine can also produce citrulline and nitric oxide by nitric oxide synthase. Citrulline can regenerate arginine by the cytosolic enzymes arginosuccinate synthetase 1 and arginosuccinate lyase. And the nitric oxide produced in this pathway is an important cell signaling molecule and acts as a vasodilator of the endothelial cells.
Hey guys, I'm sorry to interrupt. I hear you guys talk about the branch change amino acids and the arginine studies and the importance of partitioning, but I think there are more fundamental research and mechanisms that we don't know about partitioning. So John, have you think about the role of insulin in the control of partitioning of nutrients?
Maybe not enough, Carolina. Maybe you need to tell me some more.
Well, of course. Well, insulin, as we know, is one of the most important metabolic hormones in glucose, lipids, and amino acid metabolism. So before we go in more detail about the insulin controlling metabolism, we need to clarify something. So as we know, it's widely accepted that during lactation period, cows experience something called insulin resistance, which in theory occurs to help this repartitioning of nutrients to the mammary gland, but decreasing the uptake of glucose from the peripheral tissues. But if we look closely to the definition of what insulin resistant is it defined as a normal or elevated insulin levels that produce a reduced biological response. But the trick here is this insulin resistant requires an increase in the insulin secretion to counterbalance. However, what we observe in cows is the insulin actually in the early lactation is the opposite and decrease. So what we are observing is the cows are hypoinsulinemic, which might suggest that the insulin sensitivity is what is changing in the tissues instead of an insulin resistant. Okay, so we need to figure something else. Usually insulin studies measure whole body insulin rather than the insulin sensitivity in the tissues, right? So we need to know more about the liver and the mammary gland sensitivity. So there are some studies out there that indicate that the mammary gland actually respond to insulin, contrarily that we always thought about. And also it changes sensitivity of the to insulin. So for example, in studies in female rats, insulin sensitivity in the mammary gland actually increased after calving, while in adipose tissue and the muscle, the insulin sensitivity decreased. But how about cows? Right. For example, in this paper, we can observe that the phosphorylation of AKT in the mammary gland increased. So we can see that actually the mammary gland responds to insulin. I guess you guys need more proof. Well, in this paper in 2001, Masek used a hyperinsulinemic clamp, and they found that the glucose infusion rate was higher in the early lactation cows compared with the mid-lactation cows, which indicates an overall higher whole body insulin sensitivity. Another interesting finding in this paper is that the insulin plateau and also the increase in the insulin during clamp was higher in mid-lactation cows when they were compared with the early lactation one. And what it means is because during a hyperinsulinemic clamp, the cows were infused the same amount of insulin, it's possible that the mammary gland is uptaking some insulin. And I'm gonna show you a final proof. So in this paper, Sale in 2017, they used a minimal model, and then they found an increase in the whole insulin sensitivity in the postpartum when they were compared to the values before calving. So if we take all this information together, we had different gaps in the insulin dynamics, which are important to address. So first, we don't know much about the changes in the insulin sensitivity of the muscle, the liver, the mammary gland, and especially we don't know much in what happened in the different lactation stages. We don't know much about the secretory capacity of the beta cells, which determines what the circulating insulin levels are, and also the binding of the insulin receptor, which will follow by the whole activation of the insulin cascade in the insulin sensitivity, in insulin sensitive tissues. And finally, we don't know much about the role of incretins in the whole glucose and amino acid and insulin dynamics. So to fill those gaps, I'm currently doing 3 different experiments. So my first experiment has the goal to evaluate this pancreatic insulin secretion between 2 lactation stages, which are early and late. And we're doing that by maximizing the pancreas insulin secretion. So I'm using here 2 hours hyperglycemic hyperamino acid clamp, and then I'm doing another clamp, which also the glucose and the amino acid concentrations are high, but in this case, I'm adding incretins, which are known to amplify the secretion of insulin from the pancreas. And before each clamp, I'm putting these cows in 24 hours feed restriction. What we want to know is see by elevating this concentration of glucose and amino acid plus adding incretins, we will be able to determine this difference in the insulin release from the pancreas in early versus late lactation cows. And I will be using all these different measurements such as the infusion rate, the areas under the curve, and the concentration of different hormones, C-peptide, and other metabolites to find those differences. Okay, so my second experiment is designed to evaluate the changes in this whole insulin sensitivity that we were talking about, and also what is the sensitivity of the mammary gland and the peripheral tissues in, again, the 2 different lactation stages. So like in the same experiments, the cow will go for 24 hours feed restriction, and they are after they are going to a tracer baseline infusion for 2 hours. And then when I finish this baseline, what I'm doing is taking adipose and muscle tissue biopsies, which are my baseline. And after I perform in the cows on hyperinsulinemic, so here the insulin is high, and then I keep glucose and amino acid into baseline levels. And finally, when I finish about 3 or 4 hours later, I take muscle and adipose tissue biopsies, and here I'm adding mammary gland biopsies as well. So the idea in this experiment is to using the infusion of the glucose tracer to establish all these glucose kinetics and also the sensitivity in the tissues, right? So particularly in this second experiment, I'm taking jugular, milk vein, and arterial samples. So I will calculate the AV differences, which will help me to calculate the rate of appearance, the mammary net uptake, and also for differences, I will calculate the peripheral glucose uptake. And also for the tissues that I'm collecting, that will help me to determine the changes in the phosphorylation in the insulin cascade. And here I'm using techniques as a Western blot and gene expression. And finally, there are a lot of factors that can influence the insulin dynamics in the whole animal. So we went a little bit further, and we are isolating pancreatic beta cells, and we are treating the cells directly to determine these differences in the insulin secretion using 3 different levels of glucose. So if we want to see all this experiment together, we want to characterize the insulin and the insulin sensitivity dynamic in the tissues that respond to insulin, right? But the overall goal of this project is to include those findings in the development of models to predict nutrient partitioning between milk, body, and catabolism.
Okay, well, thank you very much, Carolina. So I think what you're saying is this picture I showed here was a little bit too simple. And I got to consider not just the amino acids, but kind of all of the nutrients coming in, let me call it as substrates, and how they affect metabolic hormones like insulin, glucagon, IGF-1, that kind of thing. And then because those are going to be participants in how much stimulation of utilization in the various tissues there are, that was quite clear. And then Madison and Boning are saying, well, you know, for that mammary effect, I got to look more just than mTOR, but transcription factors. And a big thing that they will do is change the number of secretory cells, you know, the proliferation and differentiation of cells that are able to carry out milk synthesis. And then, you know, Carolina, you're talking about the effects of stage of lactation. And so the lactational hormones like growth hormone, for example, that influence the pancreatic response, the insulin sensitivity of the individual tissues. But so then if this is my picture, then I think I could put some equations onto this, you know, and be able to predict what the substrate supplies from digestive absorption are. And I could use the Michaelis-Menten, you know, equation to do these various partitionings to the different tissues and effects of hormones within the Michaelis-Menten equation, that kind of thing.
Hey, John, do you think if we can predict nutrient partitioning, we could also kind of feed cows individually?
I'm sure something like that could be happening. Patty Kajerski, welcome to the presentation.
Yeah, so you know, we're talking about this insulin signaling and nutrient partitioning, and what's really going on in my mind is I'm wondering how this, you know, changes between the individual cows. So, you know what, I really view cows as individuals. To me, it's, you know, almost kind of like our having cats and dogs at home. They're all different sizes, we treat them differently, they have different personalities. And this is how I see cows, you know. They have different body weights, different compositions, different metabolisms, and ultimately this leads to different production parameters. So, you know, we might have some cows that have a higher dry matter intake, a lower milk yield, a lower dry matter intake, a higher milk yield. And I think that really shows just how different these cows are. And we've seen it in the studies, you know, but it's never really been kind of highlighted just how different they are. And going with that, it really highlights to me this big problem. We have all these individual cows, all these, you know, unique qualities of them. And essentially on the production side, we group them as this. We decide, you know, let's ignore all those unique qualities and we're going to make them all as if they're kind of the same. And we're going to feed them all the same diet based on this little blue cow here, who essentially she represents that upper percentile average cow of the herd. So let's say she's producing about 36 kilos of milk, and they'll formulate the diet just for this one cow. And then they'll decide, okay, all the rest of the cows are going to be fed the same diet. And to me, that is a problem, because that could lead to under or overfeeding. And it could also lead to excess nutrient excretion. So let's say that example that I just said, where, you know, their dry matter intake was a lot higher, but they produce less. So if that's kind of the production that they can meet and they're eating too much, they're going to, you know, excrete any of those excess nutrients that they're getting in. So, you know, looking at these cows as individuals and then seeing this problem where they're all fed the same kind of led me to that first question of, you know, can we calculate individual energy flows in dairy cattle? And could we also calculate that individual variation between cows? So this is what I did for my master's thesis. And one of the things we really wanted to do is use all the automated technology at the Elora Dairy Research Station. So there were a lot of studies, like I said, that, you can see there's different production parameters like milk yield or dry matter intake, but there wasn't anything that really took all these different automated systems and looked at these individual energy flows or individual variation between cows. And we had about 29 cows over the span of a month, and they all varied quite a bit in days of milk because we wanted to see that variation between them and then highlight that problem where they're all fed the same diet. So we used feed intake, body weight and body condition score, milk yield, and then respiratory gas exchange. So the respiratory gas exchange with the grain feed was something new because traditionally most of the research articles that you're looking at will look at methane. But what we wanted to use was the carbon dioxide production and the oxygen consumption, which we can use to kind of calculate that energy balance. And we can also use the respiratory quotient, the ratio of carbon dioxide to oxygen, to really highlight, that nutrient partitioning that we've been talking about and that macronutrient oxidation. And I'm really curious to see, like on that individual basis, how does this change individually between cows? So our main question was, yes, like we can calculate these equations, we can use the automated technology, and that really highlighted the things that we can do on a remote basis and start collecting all of this data on these cows. So to answer that first question, we looked at the variation between cows, that individual variation. And what we found varied the most between cows was our dry matter intake, milk yield, body condition score, and methane production. So essentially all of these are kind of highlighting that nutrient partitioning. This is showing where all those nutrients that the cows are getting in and how they're using them. And this is really showing that there's quite a bit of variation between these cows. So again, that makes me think, why are we feeding them all the same diet? Next, we looked at those energy flows. So first, we kind of compared our energy balance and metabolizable energy intake, and we didn't find any correlation between daily or weekly values. But the one that we found quite interesting was when we did a regression of the retained energy. So essentially that was the energy left over in the body after the production of heat production and milk production versus our metabolizable energy intake. We were able to regress that to find our efficiency of use for lactation and then our metabolizable energy of maintenance, both with our daily and weekly averaging of values. So for our daily, we found a K1 of 0.78 and then a metabolizable energy maintenance of 0.204. And then for our weekly, we found 0.74 for the K1 and then 0.188 for our metabolizable energy of maintenance. So this kind of highlighted a difference between the typical NRC values because their metabolizable energy of maintenance is about 0.125, and then their efficiency of use for lactation is, I believe, about 0.64. So this could potentially mean that our cows are a little bit more, feed efficient now. But also that metabolizable energy of maintenance being a lot higher could be due to individual attributes as well. So this makes me really curious to see how we could account for these using individual diets. And then lastly, we wanted to see if we could calculate that energy balance. So essentially our observed energy balance was the one we calculated using automated technology on farm. And then our predicted was calculated using the NRC equations. And we did find a good correlation between the two. And this really highlighted that we can calculate these individual energy flows of cows. And this can be very helpful in terms of formulating individual diets, because this way we can help to, catch before that negative energy balance in early lactation becomes a little bit too severe or too prolonged. And that way, we can help minimize any loss of production or production costs. So once we were able to do all these things and calculate that individual variation, those energy flows, it really made me think, how can we do better with these diets? So the next step in my research, hopefully we'll be able to kind of compile all this information from one cow. Essentially, we could make a virtual cow herself, use all that information that we've already kind of, validated that it does work, and then put it into a computer model. And hopefully this could help to create Snowflake's custom diet here. So I don't know about you, John, but I think like this would be totally cool and very much the future of dairy farming.
Okay, let me take what you've said and apply it on the amino acid side. You know, if you can do individual energy balance and energy flows on cows, like maybe we could apply similar things to figure out how much amino acid to supplement. So you see it's something like this, you know, you got a ration that you're feeding to cows, and then you're making these automated measurements of dry matter intake and milk protein yield and change in body weight, CO2 output. And we could take the dry matter intake measurements, so the actual observed DMI and the observed ration composition, and we can plug that into something like the NRC model to predict what the essential amino acid absorption is for that individual cow. And that from the absorption and, you know, in that partitioning model that you guys guided me towards, then we can make predictions of actually the same things that we're observing in the cows. So we can change the parameters of the model until the predictions and the observations for that cow are equal to each other. So this might be like Patty's weekly numbers, estimates of things. And so we can make, you know, run an optimization routine and generate that virtual cow that Patty was talking about. So now we got a copy of each individual cow in the herd. And if we've got, you know, the ability to predict milk protein yields and the ration composition, well, we can do things like efficiency of nutrient capture or return over feed cost just with the virtual cow, which allows us to do another optimization routine, which is to change the composition of the ration and find out which one, you know, gives us a maximum return over feed cost or combination of these various, you know, outcomes that we want to see, and then feed that ration to the cow and get the new automated measurements that we just keep every week running these two optimizations and always having the most updated virtual copy of each individual cow in the herd and keep feeding her that way.
Hey, John.
Matt Wells.
Hey, how's it going? I'm all right, thanks. I have a question though. So that modeling stuff's all pretty cool and all, but it sounds rather complex. I was wondering if there could be a different approach we could take for amino acids that might be a little simpler.
I'd love to hear about it.
Like you were saying, we use for amino NRC, and
I don't know if it's me or Matt who has died.
I think it's Matt.
Yeah, I think it's Matt.
oh, am I back?
Yeah, you're back.
Can you still see the screen then?
Yeah, we can, totally.
Okay, rural internet guys, so
yeah,
hopefully that's last.
It's a reality.
So like I was saying, so yeah, the 2001 NRC estimates the flow of each essential amino acid to the duodenum in grams per day. And it's pretty accurate in this regard, but they only define the requirements for methionine and lysine as 2.4 and 7.2% of metabolizable protein. And by defining the requirements as a proportion of the protein rather than in a grams per day figure, that really limits the ability of us to supplement our diets with essential amino acids that would allow us to feed less protein and improve our return over feed cost. Instead, the NRC predicts milk production as a function of the protein supply, and it assumes that after the cow's maintenance requirements are met, that there's a constant efficiency of conversion of metabolizable protein to milk protein of 67%. And we know from the literature that this is a large overprediction, that typically the nitrogen efficiency is between 25 and 35%. And as we increase the amount of protein we feed, the nitrogen efficiency will decline. So what we see in the NRC is that there's a large overprediction of the milk production when we feed cows near the requirement. So we're feeding more protein than what the cow can actually use. So talking about plasma profiles now, so when we do an experiment, we often look at our plasma profiles after the fact to help us interpret our results. And what we notice is that in deficient diets, the concentrations of the limiting amino acids are going to be fairly low, and that their relative concentrations will increase in response to either supplementation or an improved diet. So in this study by Broderick from 1974, we see that as cows go from a 9% crude protein diet to an 18% crude protein diet, concentrations of methionine, lysine, and valine increase, whereas the relative concentrations of the remaining amino acids remain relatively constant. So from that, we can assume that methionine, lysine, and valine were limiting at the lower levels of protein. But this is all after the fact. So after we've given the cow a treatment that we think will help improve their production. So my thought is, what if we could base our supplementation off of the profile instead? So what if we took a blood sample from a cow, ran it through our UPLC, looked at the results, and from that, tried to figure out which ones were limiting, and then from that, developed a supplementation for each cow of the amino acids that are going to help her produce more milk and utilize protein more efficiently. So my proposal is we use a target concentration for each essential amino acid seen here on the right and compare the blood samples from each cow to the reference to identify which 3 amino acids are most likely limiting. So to test this out, I used 12 dairy cows at the Elora Research Center. They were fed the standard diet, so that contained 15.4% crude protein, and it was a 3x3 Latin square with 10-day periods. So cows received jugular infusions for 4 days of either saline, the 3 most limiting amino acids based on the NRC, so that was 17 grams of threonine, 32 grams of valine, and 38 grams of lysine per day, or a combination of the 3 most limiting amino acids based on that cow's blood profile. We call that the blood-based treatment. And then this is just an example of how we calculated the amino acids that the cows on the blood-based treatment received. So in this cow, we see her Pro4, and notice with this cow that valine, leucine, and phenylalanine were the furthest away from the reference targets. So those would be the ones that we would supplement. Then we just use solver and some equations to predict how much of each amino acid we need to supplement so that they would all be the same amount off the target and sort of all equally limiting or met. So just the timeline for each period is that on day 3, blood samples were collected from the cows on the blood treatment group, and we ran those on the UPLC that night so we could identify which amino acids they would be receiving. On day 6, we inserted the catheters and began the infusions. On the afternoon of day 8 to the morning of day 10, we collected milk samples for analysis, and then on day 10, we collected blood samples from all the cows and ended the infusions. So the results, so just in this colorful table on the left is the amino acids that each cow received when they were on their blood treatment. So of note is that all of them received leucine, 9 of them received valine, and 6 of them received phenylalanine. But to the point is that we found that both the NRC and the blood treatment reduced the protein content in milk, so it went the opposite of the way we were hoping to see. And it's not entirely clear why this might have happened, but needless to say is that in all likelihood, the wrong amino acids were being supplemented. So room for improvement, obviously, for someone else who might want to look into it. One is that they might use fresher cows in their experiment. So 8 of the 12 of the cows I used were past 150 days of milk. And we know that cows earlier in lactation near peak are going to have greater demands for amino acids and are more likely to be deficient and therefore respond positively to them. And then secondly, so we sort of treated all 9 essential amino acids as like equally important, which from the literature, we know that that's not necessarily true, that there are certainly amino acids that are more likely to be limiting than others. So rather than a static target for each one, maybe we use target ranges, and that can account for the variability in concentrations we see between different amino acids. And then we better prioritize them based on our knowledge from the literature. So going back to my example cow, we noticed that after the first 3, that methionine was also fairly off target. And based on the literature, we know that methionine can often be limiting in dairy cow's diet. So maybe it's more important to address that ahead of maybe something like valine or phenylalanine, so that should go first. So there are definitely more steps that need to be taken so that we can better utilize the blood profile to help inform our amino acid supplementation decisions.
Very interesting approach, Matt. And yeah, there are next steps still to go basically on all of this stuff. you know, for this ultimate problem of being able to predict the milk component yield changes. But today was like the best office hours I ever had, with all the people coming in and teaching me all this stuff. And so the next steps, you know, were presented by Boning and Madison and Carolina on model development and things we need to consider there. And then various ways that we could implement this knowledge once we've got it incorporated into a model. So Patty and Matt both with the individual supplementation strategies that are a little bit different from each other. And I want to also acknowledge other people in the lab that are busy or you know, weren't part of the presentation here, but Jillian is our work study student. Juan and Amanda are grad students that are busy at the moment with some other activities. Michaela Chalmers just graduated this past year from the group, and Julie is always an important member. So I'll take questions, or we'll all take questions from the audience.
Thanks, John. Nice talk. Any questions for John? You can put them, you can type them in the comment section, or I think you can just speak up too.
Yeah.
I'm good at speaking up, Jen. It's John Metcalf. So I mean that was great fun for me. But actually, I had a question on well, I had lots of interesting thoughts, but the question is for Carolina's stuff. And I'm interested in the incretins because I'm not really familiar with those. But the concern I had was, how can you be certain that infusing incretins won't basically overtax the pancreas, and so that if you get a flush of insulin coming out and there's not time for the pancreas to synthesize more, how are you going to explain that in the results?
Well, that's a good question. Thanks for asking. Well, we don't know yet, because we actually don't know very well what is the role that they are playing, right? But what we are trying to do is, just because we are infusing the incretins on top of glucose and on top of the amino acids, what we are going to see is how these incretins are reducing or increasing the glucose infusion rate and the amino acid infusion rate. But that way we're seeing, like, you know, if actually the pancreas is responding to that, like, heads up that the incretin does. So pretty much is like what we are trying to see, if that answered your question.
It does to an extent, but how do you cope with the insulin clearance rate if there's a change in the insulin clearance rate? Because obviously what you're seeing in peripheral blood is only the balance between secretion and clearance. So how do you cope with that part?
Well, I think because we are trying to aim a steady state, right? So what we're trying to do is just like keep increasing glucose and amino acid to our target concentration, and then we're aiming to reach that steady state. So we are taking so many samples during those hours that I think we will be able to see those changes in dynamics. I don't know, Johnny, if you have another thought about the question.
Yeah, so I guess, yeah, degradation of the insulin could be part of it, not secretion of insulin. And actually the insulin infusion experiment, so the second part of what Carolina's doing, will probably give us more information about that. You know, if the same infusion rates give you different concentrations, it's likely to be the degradation that's causing that difference. So it is actually, those two experiments are on the same cows. They're just separated by a week or something like that.
Yeah, so, and also, yeah, that the first experiment, we are not infusing insulin. We are only infusing glucose, amino acids, and incretins.
Okay, so that'll give you more of a control.
Yeah, exactly.
Yeah. I obviously missed that in the presentation, which was really good, by the way. I did my PhD on euglycemic clamps in lactating sheep, so that's why I'm interested.
Yeah. Oh, that's awesome.
Thank you.
Yeah, my lectures on insulin sensitivity always start with your stuff on cows, John.
Oh dear, isn't there anything better, John?
Well, it was the first one. Isn't that good?
good to be known for something.
Yeah.
I have a pretty simple question for Matt based on his work. It's Kate. Hey, Matt.
Hey.
Hey, I was wondering, out of interest, I'm pretty sure that you use plasma amino acids as your indicator of amino acid status, and I was wondering if you considered whole blood and whether that might be more representative of intracellular amino acid status in ruminants. We find that for some amino acids, whole blood is preferable in monogastrics.
No, we didn't consider that, so I can't really speak to that because it was never really a consideration for us. But if you have anything of interest on that, you'd be more than welcome to send it to me because I'd be very interested to see it.
Well, we can support that. We do whole blood amino acids now, so we can support it. I'm just not sure if there's a species difference among whole blood versus plasma for a lot of those amino acids.
You know, Kate, that was a question when I was in grad school. So, you know, John can talk about his days and I talk about mine. That was a question for us about the uptakes from the blood by the mammary glands is, do the red blood cells actually contribute some free amino acid for that? And so we did plasma and whole blood AV differences of the amino acids, like not just me, but Mark Hannigan was there at the time. And so, you know, we were pooling our samples doing that. And you know, so that was a flurry of activity for a few years, and we all went back to the plasma amino acid concentrations. I think the extra work of doing the whole blood, it just didn't end up being worth it, you know, in terms of giving us extra information. So
right, probably only valid for a few amino acids, not for all of the amino acids. So it's pretty specific.
Right. Yeah.
And then I had one more question on the amino acid, on your amino acid work, Matt, which is whether you had considered looking at clustering the amino acids in kind of shared pathways, shared transporters, so that you can capture potentially ratios that might alter tissue uptake of the amino acids and then tissue metabolism of the amino acids.
Yeah, so it wasn't really something we considered beforehand, but after doing it and looking at sort of the results, it's definitely something that I'm, wondering about for, if you were to do it again, because, like in mine, we saw that a lot of the, like, with both treatments, there was quite a bit of the branch chain, so it could be a case where, like with Rochelle's past work, where maybe that influenced or drew the amino acids away from the mammary gland and, like, towards muscle or something. Which is obviously not what we were trying to do. So like trying to consider the effects of supplementing an amino acid, but we were mostly just trying to keep it simple for the experiment. So obviously maybe we need to go more complex in the future, or not me, but someone else.
Okay, Matt's apparently not doing it, John. Thank you, Matt.
You're welcome.
Hi guys, Eduardo here. I have two questions for you guys, but first, wow, what a show you guys put it. I really enjoyed the presentation. That was great. My first question was related to the branch amino acid work. I think was John that showing the redistribution among the different pools. If that was done in late lactation, early lactation, and if you expect any differences there. And my second question, John, already, then I'll stay quiet here and hear what you guys have to say. But my second question is, especially early postpartum, when we do changes, like it can be in the diet, or it can be related to health, or even frequency of milking. When you do those changes and then you remove changes that cause differences in milk production, and then you remove that intervention, very often you see carryover effects of that intervention. So I was just wondering if you guys can link all the discussions you guys have with carryover effects of intervention, like
Yeah.
is it transcription factors? Is something more permanent that is happening in the mammary gland that would, even though when you remove that stimulus or whatever it is, you still see differences in milk production for a while, sometimes for a long periods of time. So I was wondering if you guys have any ideas related to that.
Yeah, interesting questions, Eduardo. So to the first one about the stage of lactation of those cows, I think they were around 100 days in milk, something like that, 70 to 100 days in milk. So a little sort of early to mid lactation. And yeah, I would expect to see different responses at different times. And actually, I even wonder if using branched chain amino acid infusions or something like that is a good way to characterize what the partitioning capabilities could be. You know, like with Carolina stuff, we're doing insulin and amino acid infusions. And it'd be interesting if the different amino acids have different effects on that kind of partitioning and interacting with stage of lactation. That carryover effect of whatever things happening in early lactation, my first guess, I'd jump to the mammary cell number, the milk secretory cell number, and things that are happening there, the activity per cell maybe, but more like about the proliferation, differentiation, and cell death, and taking time to recover there and get back onto a trajectory, you know, a lactation curve trajectory kind of thing. So I think that's probably one of the more, it wasn't mentioned here at all, but like if we want to know how the number of cells changes in the mammary glands, we got to know how does any organ know how big it is and that it's big enough? You know, like all of the organs kind of follow that physiological workload thing of like, if there's more work for the organ, it gets bigger, and if there's less work, it gets smaller kind of thing. And so how do the mammary glands know that they're the right size they need to be for the level of nutrition and stage of lactation and all that stuff? I think that's a fascinating topic to get into. How does the liver know it's the right size, or the kidney, all that stuff? And I think it's in there. That's where I'd look.
Time for one more question, one final question. Any final questions for John or his group? Okay.
Okay.
Well, we're at that time. Thank you very much, John, and to your lab group for a phenomenal talk. Really nicely done. Enjoyed that one a lot. Thank you all for coming. Just letting you know, our next speaker for our next lab group for the CNM Nutrition Seminar Series will be next month, and it's going to be Elijah's lab. And they are going to be talking about integrating breeder and early life nutrition for enhanced poultry production. So stay tuned. We'll send out the meeting invite for that. And as always, please feel free to forward those meeting invites around. Thank you guys very much. Have a great day.
Thanks, Jen.
Thank you, John.